FRQ or a multiple choice question, you'll talk about tagged genes. A researcher has tagged a gene. I just wanted to show you what a tagged gene looks like. It can actually glow. So here a gene is tagged. The reason we see it on both sides is because this is a duplicated chromosome, right? It's in the G2 phase, or it's in the mitotic phase, right? They've already condensed, and so we've tagged this gene, and it's got one on each of these kind of the arms that we have here of the chromosome, just to kind of give you a picture. So when somebody says they tagged gene, you can go straight to this picture. Yeah, physically, I can see it when it flows like that. So this is actually fairly short lectures. We're going to talk about how we figured out that genes are actually connected sometimes, okay? And they're connected on chromosomes. Because remember that where Mendel left us, Mendel had said, well, we have these particles. This is where the particular matter of inheritance came from. We have these particles, and all of them segregate independent of one another, right? And so we can track different traits, different characters, is what Mendel called them. We can track these characters independent of everything else. That's when he got this law of independent assortment. And again, we now know that based on what he was looking at, all of his genes actually didn't assort independently. He kind of fudged some numbers in order to make the math work. But at the time, we had no idea that DNA was a genetic material. Okay, we didn't know the chromosomes were in the nucleus or how they were working or what they were doing. And so Mendel's, I guess, science background was much more limited than what we have now. And so we're going to remember just a couple basic things before we get too far. Chromosomes and genes are both present in pairs in somatic cells. Homologous chromosomes separate, and the alleles segregate during this process of meiosis, making germ cells haploid. Fertilization restores that pair condition for chromosomes and genes. Okay. So we can see the chromosomal basis for Mendel's laws, the law of segregation and the law of independent assortment, if we look again at what's happening in meiosis. Okay, you've seen a very similar version of this diagram before when we learned about meiosis. All we've done now is added the peas. Okay, but the diagram itself is the same one that we had several chapters ago. And so all this is saying is, well, we went through meiosis and we went through fertilization. We made our gametes. Okay, and then now we're going through meiosis here again. And when these are going through meiosis, notice that in this case they're all the same at this point. Okay, at metaphase one of two things can happen. And we see that here. We see that how these chromosomes line up on the metaphase plate could be this direction or this one. So the cells are committed to going down one of these paths or the other. And we see that that's going to be our law of independent assortment. How they assort on that metaphase plate is dependent on them, right? Not dependent on how any of the other chromosomes assort. And then we have the law of segregation, and we see that here. We're going to separate the alleles as we move through here, so that we have now produced our gametes, and they are a combination of both maternal and paternal traits, okay? And each one has only one copy of the allele, okay, of the gene. So I annotated this a little bit further with the text that's like on the sides here. So the law of segregation is just saying you only get one copy of each allele from each parent, and you have absolutely no control over which one you get. In contrast, over here we have independent assortment, and that's contributing to genetic variation because we don't all end up with the maternal or the paternal contribution. We have this mixing of both the maternal and the paternal contribution. So the first solid evidence that genes existed on chromosomes came in the early 1900s from a guy named Dr. Morgan. Dr. Morgan was a geneticist, but he actually didn't believe much of Mendel's work. He felt like Mendel wasn't a true scientist because he was a monk, and that Mendel had spent a lot of time collecting a lot of data, but he just didn't have the background to be making the claims that he was making. And so Morgan didn't actually set out to disprove Mendel, but he just didn't believe what he had to say. So Morgan worked with flies, and the reason that the flies were a good choice for him is because flies which happens to me like in peach season when I buy a box of peaches from the guy on the side of the road. And I mean, you may as well move if you get fruit flies. Getting rid of those little dudes, it is such an undertaking. I hate them. They are prolific breeders. They live about 2 weeks, which seems like years to me. But every 2 weeks you get a new generation. And each generation is literally hundreds of offspring. And all of those babies make for some really good statistics, right? Because the more babies you have, the bigger your N, the more likely you are to get significance. This is good news. So the flies are great. Well, so something happened. Morgan had been working with his flies for years. Okay. One day, a grad student walks into his office, very timidly, as grad students tend to approach their advisors. Says, Dr. Morgan, please pardon the interruption. I have something unusual you should see. Dr. Morgan, in my head, he's mean. I have no idea. What? What do you want, you lowly graduate student? I have a fly that's got wide eyes. All right. You've been working with flies for a decade. You've never seen anything but red-eyed flies. You've got a wide-eyed fly. It's a little boy. And he's going to live 2 weeks. Pressure's on. Right? Because in 2 weeks he's dead. You've never seen one before. Probably not going to see one again. What do you do with the white-eyed fly? Joseph, I can't help you right now. What do you need? Oh, I can't help you right now, sweet pea. I'm teaching. Sorry. Miss Ham, we'll be back tomorrow. What do you do with the white-eyed fly? You give it a lady friend. You give it a lady friend and you hope he makes more white-eyed flies. Right? You've never seen one before. So we got to figure out a way to make this happen, right? So he gives it a lady friend. And now he's really interested in what Mendel had to say. Because all of a sudden, he wants to know how to get more wide-eyed flies. And what did Mendel teach us? Mendel taught us that the F1 generation isn't real insightful. What's insightful? The F2 generation is insightful. So all of a sudden, Morgan really cares about Mendel and his work. But we need a little bit of vocab. Okay, we need a little bit of vocab. Okay? Well, every fly we've ever come across had red eyes. We're going to call that the wild type. The wild type is the normal. Well, how do you get from red eyes to white eyes? You had a mutation, right? That's how we get new alleles. We get new alleles by mutations, right? And I think now that we've covered this whole understanding of protein synthesis, I think you can see how just a very slightly different protein can get us from red to white. Very slightly different protein. Doesn't take much. Okay, very similar to brown hair and red hair, right? Just a small change in an A and a T and a C and a G. So we call this one the wild type, and we call this one the mutant. The trick is, we don't know which one is dominant and which one's recessive. We have to keep that in mind, because remember we said whatever's dominant is going to win in like the little sword fight in the cells, but just because it's going to win the sword fight doesn't mean it's common in the population. Right? We talked about with you know, the polydactyly and the achondroplasia, right? That just because it's dominant doesn't mean it's common. So we have the mutation, we don't know if it's dominant or it's recessive. It turns out in this case, red eyes is dominant to white eyes. But I just want to make sure you understand that it doesn't always happen. The mutation could actually be the dominant. Okay, the mutant could be. And so for that reason, we don't actually have a capital letter here. So we have wild type is the W and the plus. I remember this as like it is a plus, but I think of it as a T and it's wild type. I don't know. And then the W without the plus is going to be your mutant. Okay? But notice we don't have a capital in the lowercase because we don't always know. So like I said, Morgan, very interested in Mendel all of a sudden. Like, all right, so I took my wide-eyed male, I gave him his lady friend, and all of their babies had red eyes. But Mendel had taught us that the interesting stuff happened in our F2. So Morgan did not get sad about his lack of white-eyed little dudes here. So he let all of the brothers and the sisters here, and they're flies, so it's okay. All of the brothers and the sisters here mate with each other, and sure enough, he got more white eyes. But he noticed something bizarre. So he's got all these flies, some of which are white-eyed, and he starts putting them on his microscope, or his graduate students do, and turning up their skirts, and he finds out, sure enough, every single one of those white-eyed flies is a boy. And boy, that's kind of odd, because he did this a lot, right? Lots and lots and lots and lots and lots of flies, and never got a female in the F2 generation. But then he let the brothers and sisters down here mate in an F3 generation and got some white-eyed girls there. So what's going on? What's going on? Well, you know the answer to the question because, well, the name of the lecture here is chromosomal behavior. This is the first evidence we had for two of Mendel's particles moving around together. This is where our first theory of chromosomes came from. They call them chromosomes, but we didn't realize it. But Mendel had said each of these things was a particle. Whether you're a boy or a girl is a particle. Whether you have red eyes or white eyes is a particle. And they're all independent. And Morgan's saying, you know what? I don't know that that's the case. And now Morgan, he's a fan of Mendel. Because Mendel was onto something. So, what happens? So Morgan figures it out. Well, I got a mutant. And let's be honest, he's been working with literally hundreds of thousands of flies. Mutations, as we learned when we were talking about synthesis, aren't that uncommon. It makes sense that at some point he would get a mutation. That mutant happened to have white eyes. So, when he mated them, fortunately for us, flies, girls are XX, boys are XY. That's not true in all organisms, but it works out in flies. And the Y looks kind of like a crochet hook to me. It's kind of a bent little molecule. So it's easy for us to keep track of. But in essence, what he found is that, or what he determined, was that when gametes are made here from the female, they're all the same, right? Because these two are identical with regard to this eye color business. And they're both X's. But of course, Dad over here can produce two different gametes, the X and the Y. Okay? And the X here, when we match up the X and the X, we get a female, but that was wild type. Because it turns out in this case, wild type was dominant. But over here, we have the wild type from Mom and the Y from Dad, again, red eyes. That is what Morgan found. In that F1 generation, we just had a bunch of red-eyed flies. In the F2 generation, though, well, we let the brothers and the sisters mate, right? So the sister makes her eggs, and we end up with these two, red eyes, white eyes, and then Dad, red eyes, and then Y, boy. Okay, this is what we have. So when we go through our Punnett square, in and over, in and over, in and over, what do we end up with but all of our girls, here are girls, red eyes. Now this one we're going to call carrier. We got that word last time. We got carrier word last time. She's got this chromosome that's got the white-eyed allele on it, but because it's recessive, it's not expressed. But our boys are 50-50. So when we're talking about sex-linked things, as this is, when we're talking about sex-linked things, when we talk about proportions of progeny, we have to be very particular about, are we talking about half the progeny, or half the boys, or half the girls, right? We have to be very specific in our word choice. So then, here's a quick challenge. I just want you to put a star next to this, so that you can come back to it later and prove that these numbers make sense. What would we expect the progeny of an F3 generation to be, where the flies were allowed to mate freely, where the boys and the girls could all mate? Who has white eyes? And the answer to this I have here. In an F3 generation, a quarter of the males will have white eyes, and an 8 or 12.5% of the females will have white eyes. And I want you to try to be able to show this with Punnett squares. So the supposition is that everybody can mate freely. So there's 4 different possible matings. We have this female with this male. Well, who's going to have, I mean, she's got wild type. So all of them are going to have what color eyes? Red. No question, right? No question. Okay, so no matter what happens here, all red eyes. And then what about here? This female with this male, what do we get?
Red eyes.
Are all of them going to have red eyes?
Yeah.
Yeah, no question. So for these 2 Punnett squares, right off the top, a whole bunch of red eyes. Things get a little different here. Because if we do this mating, for the female, we could have this. What color eyes?
Red.
Or this. What color eyes? Again. Okay. For the males, what color eyes?
White.
White. What color eyes?
Red.
Red. So here, all the females but half the males, okay, have the red eyes. And here's where life gets interesting. What color eyes in our females? White. Males, white. Females, red. Males, red. Okay. Make sure you can go through the Punnett squares actually on paper and arrive at 25% and 12.5%. Okay, we just did it orally. But make sure that you can show that, because unfortunately your test is not oral. Right? You're going to have to write that out. Okay? But when you let them mate freely, that's when we start to find those female white eyes. But it took all the way to the F3 generation in order for that to work out. So in essence, for a male, a single copy of that mutant allele will confer white eyes. Because for a male, it doesn't matter what that little boy fly inherits. No matter what's on that X chromosome, he's going to express. Because he's only got one copy of that gene. Dominance recessive doesn't matter, because he's only got the one copy. Morgan found this correlation between a trait and an individual sex, and that provided support for this chromosomal theory of inheritance. Okay? That's where it came from. What does that mean? A specific gene is carried on a specific chromosome. So Mendel told us that all of these things are moving around independent of each other, and Morgan said, not so fast. Sometimes they do move around together. Okay? Sometimes they do. Genes are located on sex chromosomes exhibit very different inheritance patterns than the ones on autosomes, simply because in males, they only have one copy of that X. So we're going to talk a little bit about humans now that we've learned about flies. So in humans, this is an X and this is a Y. We call them a homologous pair, even though a homologous pair by definition codes for the same genes in the same place. Obviously, if we have a big one and a little one, that's not really true. But there is actually a small region here that is homologous to the genes here. And that's going to allow them to pair up during this process of mitosis and meiosis as necessary. So, just like other homologous pairs, the sex chromosomes separate during meiosis. So in a female, each egg gets one of the 2 chromosomes, X chromosomes, and in sperm, half of them get X and half of them get Y. So in humans, anatomical signs of sex, male or female, begins at about 2 months into pregnancy. I don't mean 2 months after birth. For the most part, we know if you're a boy or a girl when you're born. But 2 months old of pregnancy is when these signs of sex begin. Prior to then, the gonads, the sex organs, are generic and can turn into either ovaries or testes. What determines that is whether or not the Y chromosome is present. That teeny tiny Y chromosome only contains about 78 genes. Okay, there's not a lot on there. But one of them is a doozy. It's what's called the SRY gene, that stands for sex-determining region of the Y gene. The SRY gene codes for proteins, not just any proteins, transcription factors. It codes for transcription factors that then regulate gene expression on all of the other chromosomes. So default for an embryo is to be female. But if that SRY is present and those transcription factors are transcribed, then all these other genes and all these other chromosomes begin to be produced. Okay. So a gene that's located on either the X or the Y is called a sex-linked gene. Those that are on the Y are called Y-linked. Obviously, only males can have things that are Y-linked. But those that are on the X are called X-linked. And the fact that males and females inherit a different number of X chromosomes means that our pattern of inheritance is very unique compared to things that are actually on autosomes. While most of the genes on the Y chromosome determine sex, the X chromosome contains genes for a lot of things that are not related to sex. First, we're going to look at color blindness, and then we're going to look at your ability to have your blood clot, which is kind of important. Both of those are traits that are on the X chromosome. So, if an X-linked trait is due to a recessive allele, a female will only express it if she's got two copies. She has to be homozygous recessive. The trick is for a male, males are what we call hemizygous. Hemi means half. Hemizygous for everything that's on the X, because they only get one copy. So because of that, far more males than females have X-linked recessive disorders, such as color blindness. So when we look at this, here is how we represent individuals when we're looking at sex-linked traits. So we don't just use capital letters and lowercase letters, we actually put the X's and the Y's on here so that we keep track of the fact that we're actually moving around in higher chromosomes. But in this case, we have a normal-sided female married to a colorblind male. They choose to have children. Normal-sided mom can only give a normal X. Dad will give colorblind X or a Y. So when we complete our Punnett square, all of Dad's sons, all of their sons, will have normal vision and no evidence of colorblindness at all. But their daughters will all have one copy of that gene, and we'll call them carriers. Okay, so that's why we have a kind of a pink shaded box. They're carriers there. So in this case, a colorblind parent does not mean you have a colorblind child. However, in this case, having two normal-sided parents could lead to a colorblind child, because let's suggest mom is a carrier, dad has no evidence of the disorder, but dad's producing sperm, mom's producing eggs. Okay. If you get the normal X from mom, you're good to go. But if you get mom's disorder X, well, the girls are the carriers and the boys are colorblind. Okay. It is possible for a dad who's colorblind to marry mom who's a carrier and still produce a son that has no evidence of the disorder, if dad gives the Y, as he must, and mom gives the normal-sided gene. But that's how you get females with color blindness. If you're a female and you're colorblind, your dad had to be colorblind, and your mom had to be a carrier. It's the only way to get that, which is much more uncommon, and that's why finding females who are colorblind is actually quite rare. Okay? I'm told, just in the last year, you can buy glasses that will adjust light and allow, for some individuals who are colorblind, allow them to actually see color. But up until very recently, there was nothing that could be done about this. And so the trickiest part, I'm told, was driving. Because most people who are colorblind are red-green colorblind. And so being able to see the stoplights is hard. Okay? I am to understand that the red is always at the top, and the green is always at the bottom. The trick is on the sideways ones. Apparently there's not consistency with regard to the sideways ones, which I've never, ever paid attention to. But that's where life gets a little bit tricky. There you have it. All right, what's this? pedigree of, can you see it? The hemophilia in the royal family. So being colorblind isn't really of any great consequence, but not being able to have your blood clot is a much bigger deal. And so Victoria, Queen of England, who lived in the 1800s, was actually a carrier. We're not sure where she got it from. One of these two, obviously. But we're not certain where. And so she was a carrier for the disorder, and they had a lot of kids. Okay. And that whole collection of kids, we got a carrier Nellis, Leopold, well, it's a boy, so you either got it or you don't. He had it. And then Beatrice, I believe. I think that's what that says. I can't see it. I think that says Beatrice. Beatrice was a carrier as well. But of course, back in the 1800s, there was no test for this. We couldn't figure out if you were a carrier, you were just healthy, or you weren't. So Beatrice married off in Spain, and so generously gave her disorder to the Spanish royal family, where it was actually a much bigger issue than it was over the English side, just out of remarkably bad luck, to be honest. But you'll see that the males, for the most part, don't have kids. You don't survive it. You don't survive hemophilia. We have one exception here. Leopold managed to survive long enough to have kids, 2 of them, but he did give that to his daughter as a carrier, right? And of course, we didn't know that these sorts of things existed. And it just got spread through the royal family. So here's our current royal line. Over here. Here's Prince William and Princess Kate. And I need to update this so that I can add, what's his name? Henry, Charlotte, and what's the boy's name? Is it Henry? No, it can't be right. I don't remember. Harry. No, that's him. George. Charlotte and George. There it is. Charlotte and George aren't on here. And so our current royal line is doing just fine. I will highlight to you that it seems apparent that Queen Elizabeth and Prince Philip missed out on the lecture last class, which included the topic, why you should not marry your cousin. As you see here, they're totally so, well, there's that. Now, I do believe that they are 4 generations removed, right? But they do have, they both come from right here. So that would have been mom, grandma, great-grandma. So they have the same great-great-grandparents. And these are 2 people who are still alive. But this is the current queen and her husband, Prince Philip, have the same great-great-grandparents. So there you have it. And they're a lovely couple. It seems to have worked out for them, right? Their kids seem well-adjusted and extremely good-looking and wealthy, so there you go. But lucky to not have hemophilia, because that really, we got lucky with regard to the hemophilia, because it could have just as easily popped up here or here. But for so long, royals only married royals, and if you were of royal blood, that was a good thing, and in this case, royal blood was not a good thing. Quite literally, not a good thing, not a good thing. That's kind of how it always done. It's an excellent thing. So we're going to remember the Dean's Code for proteins. But men, the existence of men, tell us that only one X chromosome is really necessary to produce the required number of proteins, since there's more sex characteristics on the X, and males seem to get along just fine with only 1 copy. So girls have 2. So what happens to the extra X? Well, the extra X is inactivated in females very early in embryonic development, right around the 100 cell stage. So a couple days old, right after fertilization, division starts, mitosis, mitosis, mitosis. And right around the time all of us girls were at about 100 cells big, in each cell, one of the X's was extraordinarily methylated, highly condensed, and turned into something called a bar body. And it's not expressed at all. And in that way, all females essentially have the same dose of X of 1 as males. Now, this happens, like I said, very, very early on. In the ovaries, where meiosis is occurring, bar body chromosomes are essentially demethylated before meiosis to make sure that they are equally passed on to your children. However, in every other structure in the body, whichever one is methylated and turned into a bar body is consistent through rounds of mitosis. We call that epigenetics, right? We talked about that before. That genes that are highly methylated and essentially turned off, maintain that through rounds of mitosis. And so what ends up happening is at that 100 cell stage, randomly in each one of the X's is turned into the bar body. And then as that cell goes through mitosis, whichever one was deactivated is also deactivated in the daughter cells. Okay? So they call females a mosaic, where you have two types of cells. One with an active paternal X, and those cells with an active maternal X. So if a female is heterozygous for a sex-linked X-linked trait, about half of her cells will express one allele, and the others will express the alternate. We can observe this in calico cats, okay, where orange is on one X chromosome and black is on the other X chromosome. Okay, you can't have a male calico. Now, every year somebody wants to argue that with me, and I say, well, you can have a male cat that has a weird coloration, but they're not a true calico, because in order to be a calico, you have to have two X chromosomes. That's not true. Okay, we have a similar condition with humans. It's an X-linked mutation that prevents the development of sweat glands. And so there are female humans who are patchy with regard to their ability to sweat. They have regions of their body where they cannot sweat. And a lot of times you don't notice it, right? Because as long as you cool your body down, I gotta be honest with you, I'm not sure I would notice if one part of my body was lacking sweat versus another part had too much. This isn't really something I pay attention to. But the disorder absolutely exists in a similar fashion, where you're just patchy with regard to what got turned on and off. So what we're going to do is watch a video called Why Women Are Stripey. Just a quick YouTube 5-minute-er.